So, good evening, everyone. Welcome to the session for this week. Today, we'll be going over the initial assignment that was given as part of the topics that were discussed in week 6. After that, we will look at the topics that were covered as part of week 7, get into some details about it, get over the lectures that were already there, go over the lectures that were already there, and after that, we'll come to some of the sample problems for the upcoming week. And we can then have a discussion session for the final week in the upcoming session. So I will first begin with just yeah. So first we'll go over the assignment for week 6. So the first question for the assignment was about actin. So we had a lot of discussion about the cytoskeleton, its formation, all of the different types of the changes that occur within the cytoskeleton, how it is involved, what is the role of each cytoskeletal element, how does treadmilling occur, how dynamic instability, etc. All of that details we have with us. So the first question is about how actin polymerizes into microfilaments using DASH, while tubulin polymerizes to form microtubules using DASH. If you remember, we had discussed dynamic instability and treadmilling for both these types of cytoskeletal elements previously, and the type of energy molecule that is involved in each case is different. In case of actin, it is ATP, and in case of tubulin, that is GTP. So if you remember how we had seen it as a conveyor belt, from one end there is depolymerization that is happening, from the other end polymerization is happening. So there's a constant cycling or a constant treadmilling which is taking place in those cases. So that is what we saw there. Then coming to the second question, actin filaments associate with DASH to facilitate muscle contraction. So we have dyneins and kinesins, we have myosins, kinesin, and myosin or dynein. So actin filaments in this case, they, if you remember the muscle contraction or the sliding filament theory, which is the basis on which muscle contraction is understood, then in that case, you would remember that myosins, which are the major component, they mediate the sliding, like the two types of filaments, the thick and thin filaments that form the part of the entire skeletal unit or a sarcomere, that was contributed by myosins. Myosins were the thick filament and the actins were the thin filaments, which together helped in sliding along each other to mediate the contraction of muscles as we know them today. So that was question 2. Then in question 3, we were asked that which of the following processes could be primarily affected by the disruption of nuclear lamins. So if you remember, we had looked at intermediate filaments as well, and lamins were part of the intermediate filaments as we knew them. So what is the impact of the disruption of nuclear lamins? So first, the proposed options are that it could disrupt the shape of the nucleus, it could affect gene expression, it could affect the condensation of chromosomes, or all of the above. So because nuclear lamins are mediating the architecture of the nucleus and the demarcation between the nuclear environment and the extranuclear or the cytosolic environment. Therefore, any disruption in the composition or any impact on nuclear lamins could also by large have an impact upon the shape of the nucleus, upon the expression of genes, because while the genetic material is condensed within the nucleus, its expression is carried out in the cytosol. And that would be mediated by not just the not just what genes are present inside, but instead what genes are being finally transcribed and finally translated in the cytoplasm. And therefore, that requires the correct transport of the mRNA that needs to be translated from the nucleus into the cytosol. So therefore, gene expression could also by large get affected by this disruption. And thirdly, condensation of chromosomes also would get affected, and therefore the correct option for this is all of the above. Then in question 4, we've been asked that about centrioles. So So centrioles, they play a role in organization of the actin cytoskeleton, polymerization of microtubules, cross-linking microtubules and actin, or attaching motor proteins to microtubules. So the correct answer in this case would of course be microtubule polymerization. You remember we had in some slides we had some extensive details about what is forming the centrosome that we know it, the centriole and the pericentriolar material from where microtubule organization center is present and from where microtubule polymerization would take place. So that is the key center from which the microtubules would originate and how tubulin would keep getting added or tubulin would get removed. And that would effectively lead to the change in cell dynamics that would help in cell division and many other functions as and when they are required. Then in question 5, we were asked that which of the following statements is true. So not all motor proteins have directionality. Certainly not true. Motor proteins do have to move in either direction. They would either move towards the plus end of microtubule or minus end of microtubules, and they would depending on which class they belong to and which type of motor protein it is that we are talking about, we can actually have several subclasses and their directionality is critical to be maintained in the long run. Then we are asked that microtubule plus end is pointed towards the nucleus, ARP23. So microtubule plus end is certainly not pointed towards the nucleus. It's a false statement. Then we have ARP23 nucleates new microtubule filaments, also not true. If you remember the additional slides that we had looked at in our previous session, there we had seen that formin and ARP23 are two major proteins that are involved in that are involved in the nucleation of actin and how they help in addition of individual actin monomers to form trimers, and then that polymerizes and forms the entire microfilament. So certainly RP23 is not involved in microtubule nucleation. Yeah. And then in question 4, in option 4, we have that microtubules are the central support in flagella. This is of course true. If you remember, we had looked at the 9+2 arrangement. We had seen how it forms the axoneme and how there are doublets of microtubules on the outside, and we have the central microtubule assembly. So all of those details were discussed, and therefore option 4 or option D would be correct for question number 5. Then in question 6, we're asked that bimentin is an example of A, motor protein, B, an intermediate filament, C, microtubules, or D, microfilaments. So bimentin, even though we did not get into much detail about it, it is a type of intermediate filament, and therefore option B would be correct in that case. Then in question 7, we were asked that which of the following statements about microfilaments is false. A, microtubules are the thickest class of cytoskeletal fiber. It is true. So first of all, this statement is not about microfilaments. This is about microtubules. They are different. And it is also a true statement. Among these three, they are the thicker filaments. Among microtubules, actin and intermediate filaments, microtubules are the thickest filaments and or the thickest class of cytoskeletal fibers. Therefore, that statement is true. In option B, we are asked that microfilaments are made up of globular actin or G-actin, and also that is true. Just like we just said, that formin and ARP23, they help in assembly of the actin monomers, the individual G-actin monomers, which together form the filament, first trimers and then the entire filament. So the second option is also correct. Then in the third option, we are asked that intermediate filaments, they bind to motor proteins. Yes, they do. They do. Intermediate filaments do not have any. I'm sorry, it was wrong on my part. Intermediate filaments do not bind to any motor proteins. Only the other two, because they do not have any role in cell mobility per se, or do not have much of a role in cell division. They are mainly for structural support, for providing the strength or in fact, the mechanical strength within the cell in different locations based on where they're present and what is their function. On the other hand, if we talk about microtubules or if we talk about microfilaments, they are associated with these functions and they are acting as those, like we talked about the railway lines on which a train would have to pass. So they would serve as those railway tracks. So in their case, the movement of motor proteins is essential and of course carries directionality. But in case of intermediate filaments, it is not true. So that option C is correct because it is the false statement. And microfilaments, they show treadmilling during cell migration. That is of course true. Treadmilling is something that we observe. And yeah, that these are the four options that were there. In question 8, we're asked that which of the cytoskeletal components lacks polarity. So polarity basically means that one end is plus, one end is minus. So this directionality or this polarity is absent in case of intermediate filaments. So we have seen that both plus and minus ends are established when it comes to microtubules and when it comes to actin, but certainly not when it comes to IS. So in this case, option A is correct, and that would be the correct answer. The homologue of tubulin, which is found in bacteria, is MORB, CRE-S, FTSZ, or there are no tubulin homologues. So we have discussed this several times in the last 2 weeks, and the correct answer for this is that it is FTSZ, which would be option 3, and that is the correct answer. And which of the following drugs inhibit actin polymerization? So I don't think we had particularly discussed this example. I'm not sure. But because we had discussed vincristine and vinblastine, and we had seen that they inhibit microtubule polymerization, microtubule assembly. So those two options are certainly not correct. So in case you are not aware of this example in particular, the first example in particular, then you can, by the process of elimination, also you will be able to arrive at an answer that would be latriculin A. So option A would be the correct answer in this case. So that is bringing us to the end of the assignment questions that were there for week 6. So as per the pattern, before we get into the questions for week 7, we'll go over the topics that were part of week 7. So for that, it's visible. So if you've, I assume everybody has already gone through the lectures. So the topics that are covered in this week have been centered around the endomembrane system. So what is the endomembrane system? Basically, if you just dissect the terms, you will see that it is endomembrane. So membrane, which membranous structures or membranes which are present inside the cell, not the cell exterior, but inside the cell. So that would, whenever we talk about membranes, it usually means the cell membrane or the plasma membrane, or max to max we think about the nuclear membrane. But certainly if you think about it, the bilayer or the membranous structures are present throughout the cells for almost many of the major organelles that we talked about. And that starts with, as you can see in this sort of digital cross section of a cell as well, that it starts with the nuclear membrane or the nuclear covering, following which we have the endoplasmic reticulum, which again is subdivided into two types, whether based on ribosomes, but based on whether there are ribosomes or there are no ribosomes, so that would be smooth or rough endoplasmic reticulum, following which we have the entire Golgi apparatus. Whenever we say apparatus, it is because it's not a singular entity by itself, like there's one nucleus and it's a composite entity. The endoplasmic reticulum or the ER is a very scattered sort of arrangement, or a scattered it's a network. It's like a both the ER and the Golgi are sort of a network. In fact, in some cases, in some textbooks and papers, you'll also find the Golgi being referred to as the Golgi network, the trans-Golgi network, or TGN. So because there are multiple layers, all of which are interconnected to each other. So both of these are not just singular entities, but rather a number of compartments that are all interrelated and interconnected. And finally, of course, you have the cellular membrane. But apart from that, we also have lysosomes and vacuoles, which are structures that will also come to much later. But the major focus will certainly go towards endoplasmic reticulum and Golgi. So what is the endomembrane system? So many of the internal membranes in a eukaryotic cell are part of the endomembrane system. Either they are present, these membranes that we refer, they are either present in direct contact, or they are like they're directly in contact with each other. Like we'll come to eventually in a few slides, the sort of parking lot arrangement of this network that I was talking about, that they are connected to each other physically by some sort of membranous network. Either that is the case, which is what we see for ER, that in the ER, if you have ever looked at a diagram quite carefully, I think you'll be able to see here as well. So if you look here, you'll see that there is the first layer of the ER is there, and it is connected to the second by sort of a little link that is connecting each of these spaces to each other. So yeah, so that's what we see there, that they are in direct contact with each other, or in some cases, and like in case of Golgi network, they look like they're independent membranous structures, but they're actually connected to each other because of some sort of vesicular pinching. And like from the first Golgi compartment, which is the cis Golgi compartment, a little bit of the membrane pinches off, fuses with the median. From the median, a little bit pinches off, fuses with the trans, and from the trans, it pinches off and goes towards the cell membrane or the cell exterior. So we see that connection being present there. So in spite of these links, these membranes have very diverse functions and structures, and in fact, these membranes get even modified during life. Now, the endomembrane system, it includes, first of all, like we were discussing, it includes the nuclear envelope. The nuclear envelope is connected to the rough endoplasmic reticulum, which is also continuous with the smooth endoplasmic reticulum. So there's a more detailed diagram here, which I think if I zoom in, it'll be visible. But yeah, you see this is the nucleus. It's like a cross section, like if you've sliced a cell right through the middle, this is what you would expect to see. So you have the nucleus present. Right next to it, we have the endoplasmic reticulum, which is present. And the nuclear envelope is directly in contact with the RER, or the rough endoplasmic reticulum. The reason being, which is something I think we discussed in our earlier classes as well, when we talked about the central dogma, where we talked about how from the nucleus, right after transcription has occurred, you want the time gap, if a gene is to be constitutively expressed, or if it has to be quickly expressed right after it is being transcribed and it needs to be translated, then in that case, you want that accessibility to be present there. And therefore, the immediate connection right next to the nucleus is the rough endoplasmic reticulum, which has ribosomes sort of beaded all over it. If you see here, you'll be able to see little tiny dots which are present. While we have looked at the structure of ribosome, and we have seen that it's a huge biological macromolecule, and it has it looks like it has two entities. It has a large subunit, a small subunit, each of which is made up of proteins and RNA. It can function as an enzyme, and the whole ribosome is a ribozyme and it comes in. But when you look at a cell at large, and you'll be able to see that the entire membrane is sort of dispersed with the structures that are present all over it. And the rough endoplasmic reticulum is right next to the nucleus, and from there it moves on to the smooth endoplasmic reticulum, which does not, of course, does not have these structures. And from there we see that there are little sort of vesicles emerging. If you can see these membranes and these proteins that are produced by the ER. So some proteins that are this is a very superficial example, but when you go into much more depth, there's an entire topic of vesicular trafficking which takes place inside the cell, which is so finely controlled, and that has to be controlled so finely that it's not something we can cover so superficially at this point of time. But for now, what you can just think of is that the ER sort of acts as the traffic police of the cell. So whenever some protein, whenever most proteins, when they are synthesized with a tag. When they're getting transcribed, translated, they have a tag which is known as a signal peptide. What you understand by that is that that is the address of the cell. So address within the cell where that protein has to go. Does it have to go back to the nucleus? Does it have to stay within the ER? Does it go to Golgi? Does it go to the membrane, lysosome, or is it a cytosolic protein? What is its job? If you think about it, there are hundred thousands, hundred millions of proteins inside the cell, right? Each of these cytoskeletal elements that we talked about in the last week, they're made up of tubulins. Those tubulins should be accessible in the cytosol so that the microtubule can either be formed or deformed based on what the need is. So its accessibility has to be within the nucleus. Versus if you think about a protein like DNA polymerase. DNA polymerase does not necessarily have a role inside the within the cytosol. It should go back to the nucleus. It should be available within the nucleus for the process of replication to be smoothly conducted out. It is possible that some proteins might not go, but if it is supposed to do its canonical job, it should be present within the nucleus. So this correct localization of the protein, that a protein which is meant to be where it is meant to be, that is carried out at the most, under most circumstances, is carried out at the ER. So after the protein has been synthesized and that tag or that single peptide that I'm talking about, or the address of the protein has been identified, the ER performs this major function under most circumstances, that it directs it to the appropriate site. I might have a slide on this. We can discuss it if that's something that anybody is interested in. But yeah, it's a slightly complex concept. The basic idea is the same. The signal peptides for each case are different, and there are different recognition proteins that identify, oh, this is the signal that's there, and therefore it carries out the correct transport to the correct site. However, for now, what we can understand is that there are certain proteins that are being synthesized inside the ER. And once it has been synthesized, it is sort of packaged in a vesicle, and that vesicle is made up of the membrane, the ER membrane, basically, the lipid membrane, basically. So what it does is that the Golgi, it pinches off from the ER. These membranes are being carried to the Golgi, and from the Golgi, it pinches off these transport vesicles and other vesicles that can give rise to other endomembranous compartments like lysosomes and vacuoles. Then you see that the lysosome in legend 4, you can see that the lysosome is available for fusion with another vesicle that might actually have to undergo digestion. So both of these compartments, they fuse, and so the enzymes that were present inside the lysosome can digest whatever needs to be digested, and the lysosome is completing its job. So this is a membrane fusion which is occurring. Then we have the transport vesicle, which can carry some proteins to the plasma membrane for secretion, that they need to be removed outside the cell. There's some protein which is being released outside the cell, so that has to go through this entire network, then has to go to the cell membrane, and from the cell membrane, it has to be moved outside the cell. And plasma membrane as well can expand by the fusion of these vesicles, and the proteins instead are secreted outside the cell. Now there are a huge number of questions that remain here. That is there no identity of the lipids that come from ER, or the lipids that come from Golgi, or those that are on the membrane? There are a lot of ifs and buts. There are many studies, many groups that are carrying out studies on these topics, and it's extremely interesting if it's something that you want to read up on. What we know for now is that lipids by themselves do not carry much of an identity, but yes, if it is emerging from a specific compartment, should it have a certain composition, or should it have a certain distinction? Does it affect where the lipid is coming from, or is it what is inside? Is it just like a box, and you can keep anything inside that box, or does that box determine where it has come from? Does it belong to some XYZ compartment? Those are all open-ended questions that certainly some of which are being answered. You can look it up if that's something that interests you. For now, what else you can also keep in mind is that the default address of any protein that is being synthesized within the ER is to go into the cytosol. So if there is no specific signal peptide or no specific address that has been attached to the protein of interest, then that protein will stay in the cytosol. On the other hand, if there is some specific address which is given to it, it will accordingly get redirected to those sites. So that was the overview of the process that occurs. So first we look at the ER. Now the ER accounts for half of the membranes in a eukaryotic cell. Like I said, it's a very extensive network, and it performs such an intensive and important job for the cell as well. It needs to occupy that much space because it is carrying out that important job. And this is not the only job that the endoplasmic reticulum has. There are other metabolic functions as well, which are taken care of by this particular compartment. And if you read more, the ER has more and more things that are getting discovered every day. You look into it. So the ER it includes membranous tubules. You're looking at membranous structures, membranous coverings, and the internal space, which is it's not an empty or not a hollow space, but instead these are fluid-filled sort of tubular structures, and those are known as the cisternae. So, endoplasmic reticulum cisternae, you can do, cisternae are basically these sort of sac-like structures which are present. They're used for Golgi as well, for DER as well. So, the ER membrane is continuous with the nuclear envelope, and the cisternal space of the ER is continuous with the space between the two membranes of the nuclear envelope. So that's why we said that it is directly in contact with the ER. And like we have previously discussed, we understand why that is important as well, and why that is the default pathway that a cell chooses to be in as well. So that's what we saw. And following that, we are seeing that the two, although connected, regions of ER that are known to us, they do differ in their structure and function. So first we have the smooth ER, which literally looks smooth. It lacks ribosomes and it looks smooth. And the rough endoplasmic reticulum, which is rough because of the presence of ribosomes, or the bound ribosomes, they are attached on the outside. They include the outside of the nuclear envelope. So you can see an electron microscopy image where you can literally see these teeny tiny little sort of dot structures which are present all over, and that is representative of the rough endoplasmic reticulum. So yeah, that is how the nomenclature, I believe, was also brought about. So if we are talking about just the rough endoplasmic reticulum and the ribosomes and their job, then what is the smooth endoplasmic reticulum doing? Is it just there for the hell of it? No, it has a different set of functions that the rough endoplasmic reticulum is not usually associated with. First of all, the smooth ER is quite enzyme rich, and like I said before, it has roles that are more towards the metabolic activities of the cell. So enzymes of the smooth endoplasmic reticulum are involved in lipid synthesis, which includes oils, phospholipids, and steroids. Some specific types of lipids are also digested, some are synthesized. It depends on what is the function of each organelle. And this also includes, when we talk about steroids, it includes the sex hormones of vertebrates and adrenal steroids as well. Now, the smooth endoplasmic reticulum also catalyzes a key step in the mobilization of glucose from stored glycogen in the liver. The smooth endoplasmic reticulum is also like this was one set of functions, but another set of functions involves that is specific to the hepatocytes, that within the liver, it carries out the movement of glucose from glycogen. Glycogen is the storage form of glucose. From there, the accessible form of glucose is brought about. So there's an enzyme that removes the phosphate group from glucose phosphate, which is a product of glycogen glycolysis and hydrolysis, and that permits the glucose to exit the cell. So this is the job, or this is the function that is being carried out within the smooth endoplasmic reticulum. On the contrary, the rough endoplasmic reticulum is involved in a different set of functions. So if you look at that image, you'll be able to see this clear. Obviously, it was the colored here, so you'll be able to see the clear distinction here as well. So it is especially abundant. The percentage of smooth versus rough endoplasmic reticulum can vary to some extent. Even though I do not have studies in my mind that I have already read upon, but yes, you can come across these papers if that's something that again interests you. But there can be a difference in the percentage of the smooth endoplasmic and rough endoplasmic reticulum. And as a polypeptide is synthesized by the ribosome, it is threaded into the cisternal space through a pore formed by a protein in the ER membrane. Just a second, some disturbance. It's just give me a minute. There was some recording issue. Should be okay now. Yeah, so we were talking about the endoplasmic reticulum, the rough endoplasmic reticulum in particular. So it is believed to be especially abundant in those cells that secrete proteins or that are more responsible for the secretion of proteins. So when a polypeptide is getting synthesized by the ribosomes that are present on the RAR, it gets threaded through the cysternal space, and this threading occurs through a pore that is formed by a protein in the ER membrane. And many of these polypeptides are actually glycoproteins. And when we say glycoproteins, it basically means proteins that are carrying some sort of glycosylation. Um, the process of glycosylation is also something that is it's a detailed process, and that is another major function of the endoplasmic reticulum as well. So I think there's another slide which sort of brings it up a little bit. But basically, a sugar think of it like a sugar tag. It's not a tag per se, the same way that we would talk about a signal peptide, not in that sense, but basically it is a sort of sugar moiety that is added to the synthesized polypeptide, and that has some specific roles. That like a large percentage of the proteins inside the cell are actually glycosylated. There are two types of glycosylation. There can be N-linked or O-linked glycosylation. There's a lot of detail that you can go into about it. It depends on which residue is getting attached with, or the residue to which the sugar moiety is getting attached. Based on that, the nomenclature will vary. So it can either be an aspartate or it can be a serine. So there's a lot of detail about it. So this whole domain is covered under vesicular trafficking and protein localization, etc. However, for now, what you can understand is that these glycoproteins that are polypeptides to which an oligosaccharide is attached, they are often synthesized and within the ER, and therefore this is another major function of this particular endomembranous organelle. Uh, Now, these secretory proteins, they are packaged into little vesicles that we talk about, like little envelopes, like lipid envelopes within which these proteins are present, and then they are carried over to the next stage and they pass through the cis-medial trans-Golgi network, and then they would be secreted outside the cell after fusion with the plasma membrane. So that depends upon what job it is supposed to carry out. Um, the rough endoplasmic reticulum is also considered to be a membrane factory. So membrane-bound proteins are synthesized directly into the membrane. Enzymes in the rough endoplasmic reticulum, they also synthesize phospholipids from precursors in the cytosol. So like I said, the ER is occupying such a huge part of the cell because it is performing such a huge I would not say it, but like a diverse range of functions. It's not just doing one type of job. Whatever jobs it is doing, they're very um, crucial for the survival of a cell, and they're actually a little different in their functions as well. So some functions include the fact that they synthesize phospholipids, which is kind of similar to what we talked about the smooth endoplasmic reticulum also, that it also is involved in the synthesis of lipids in some cases, in the oxidation of lipids in some cases. In case of rough endoplasmic reticulum, in a large number of cases, it produces phospholipids from the precursors which are already present in the cytosol. As the ER membrane expands, some parts of it can be transferred as transport vesicles to other components of the endomembrane system, which is what I talked about as the pinching, that little vesicles are pinched off from the larger organelle and then they're carried forward to whatever their um, destined location might be. So that's what we see here. And here, this is what we had seen in the lectures as well, that it has a sort of parking lot or a spiral sort of architecture. So you can see that they look like they are uh, like pancakes, one stacked on top of the other, not exactly in direct contact, but there's a link. It's sort of like a spiral. And if you pull it apart, they're all interconnected, sort of interconnected. In direct contact with each other. So you can see that this is the parking garage model of these stacked ER sheets. And you can see there's a helicoidal ramp. It's just how the structure is mentioned. And there are some ribosomes because here we talk about smooth and rough endoplasmic reticulum. So it has the membrane-bound ribosomes right in direct contact with it. And this is a 3D sort of space filling model of the same architecture to show how it looks. And if this is something that interests you, you can certainly go and check out this paper. It was published in Cell. that stacked endoplasmic reticulum sheets are connected by helicoidal membrane motors. So that was the architecture which was identified by this particular group. So it would be an interesting read. Go ahead and check it out to just get an idea about what exactly is happening. Now we'll come to this job of the another job of the ER, which is what I've been talking about a little bit. I will not get into much detail about it, but we'll at least get a holistic picture of what is present there based on just this figure. So yeah, if you see here, you will see, oh, sorry. You will see that there are a sequence of events which are occurring. The first job is that in a large number of cases, you will have some cytosolic ribosomes which are present. Some ribosomes are attached, but not all. Some ribosomes are free and they're present within the cytosol of the cell as well. So what happens is that after an mRNA has been transcribed and it has incorporated whatever modifications it needs to form the mature mRNA, what it does is that it attaches or it is taken over by a free ribosome which is present in the cytosol. And there the polypeptide synthesis begins. So translation, the process of translation begins from that stage. So if you see here, you'll be able to see that this is the polypeptide gene which is getting synthesized. And at the very beginning, what you see is a little bit of a signal peptide. It is also a protein sequence, but these are distinct protein sequences which are which do differ based on where these proteins need to go, whether it is the mitochondria, whether it is peroxisome, whether it is just staying in the ER. So depending on that, each of these you get like a table of all of the different types of signal peptides that are present. Now this signal is recognized by something else. What recognizes it is SRP, or signal recognition particle. So what it does is that it goes and it binds to this particular sequence and it pulls that entire complex, not necessarily pulls, but it brings that entire complex, that is the SRP signal peptide, rest of the polypeptide attached to the entire ribosome, which is attached to the mRNA that it is currently translating. So this entire complex is brought to the ER. And this is localized on something called as a translocon. If you see here, this is brought onto a translocon, and that translocon is it's not it's the entire complex of translocation, and that has a signal recognition SRP, or signal recognition particle receptor protein. So SRP is present with the entire macromolecular complex. The SRP receptor is present on the ER. It is present in various sites on the ER. So this entire complex would be brought and would be bound to or would bind to the SRP receptor, which is present on the endoplasmic reticulum. Upon binding, what will happen is a little bit of shift that occurs. And next step, what happens there is that the SRP binds to the SRP receptor. Okay, I'll go over step 2 as well, that when SRP binds to the signal peptide, synthesis at that point is stopped. That the peptide is not getting extended, unnecessarily extended further. At this point, what is critical, that after recognition has happened, it needs to be brought into the ER. So the SRP binds to the SRP receptor protein in the ER membrane, which is part of the translocation complex, and that has a membrane pore and a signal peptidase, or signal cleaving enzyme. So the SRP receptor, if I zoom in here, yeah. So orange is your SRP receptor, which is bound to this part of the translocation complex. And it, as you can see, it is bound to this green signal peptide as well. So this entire docking has occurred. After this, what will happen is that the SRP, or signal recognition particle, would leave after this recognition process has been carried out, and polypeptide synthesis would once again resume. So why was it at all stopped? It was stopped because this protein, it might just so happen that after the signal peptide, there are some residues which need to undergo glycosylation. That glycosylation cannot occur within the cytosolic environment. That needs to come into the ER. It needs to be hijacked, brought into the correct compartment so that the correct glycosylation can take place. So everything inside the cell, under normal circumstances, everything is so beautifully and so perfectly finely regulated that there is still so much that we don't understand and we don't know. And that is so surprising and so beautiful at the same time. So the SRP leaves, polypeptide synthesis resumes with simultaneous translocation across the membrane. So what now happens is that while the processing is or while the process of translation is occurring, it is also getting localized inside the ER. In fact, there are some concepts which suggest more translational folding. So in that case, if it is this is a slightly more advanced concept, but in some cases it can happen that if it is say a like GPCRs, if it is a 7-pass transmembrane protein, then in those cases there are um, certain proteins which are able to recognize that this particular region is supposed to be membrane localized, this particular region is supposed to be present in the cytosol, this particular region is supposed to be present on the inside of the endomembranous system. So, in that place, that entire orientation can actually occur. So these are beautiful, beautiful concepts. If you are interested, I can share some slides on it or I can recommend books on it. They're slightly difficult to understand at the first go, but once you understand how it is happening within the cell, it's fantastic. So anyway, coming back to the discussion at hand. So the polypeptide synthesis resumes and translocation is carried out across the membrane. The signal peptide still stays attached to the translocation complex. Now in step 5, the signal cleaving enzyme, which is otherwise known as the signal peptidase, will cleave it and the rest of the protein is released into the ER lumen. And this is the rest of the completed polypeptide will leave the ribosome and will then undergo folding. This entire um, this particular sentence has summarized a huge area of scientific research, which is protein folding, which has so many different angles, so many different hypotheses that there is still no one clear answer to it. Some concepts say that folding is occurring within the ER. Some are saying that the ribosome acts like a scaffold, like it gives. If this is the ribosomal surface is providing the area for folding to occur. Some say that folding begins within the polypeptide exit tunnel. Some are debate. There are open debates everywhere because folding of a protein is one of the most critical steps, as you can understand, which will determine what is the way that protein will function within the cell. You can't just it's pointless to just have an mRNA being synthesized or to just have a polypeptide being synthesized. That has to take its perfect conformation for it to be able to do its job. In fact, misfolded proteins lead to XYZ number of diseases, which are much worse for the cell than to not have um, to just have any protein at all. So these are very, very crucial steps, and there are many different angles that many different study groups look at. And maybe several years from now, we'll have one clear defined answer, or maybe we'll understand that multiple methods they operate or they function in parallel within the cell, which allows protein folding to occur the way it does. So yeah, so these are the different steps that take place within the cell. And here we had another video, I think, that was present. So that completes the structure of or the function of endoplasmic reticulum. The next is the Golgi. So its job has been summarized into 3 words: that finishes, sorts, and ships cell products. So kind of like Amazon, but not exactly the same. So what it first does is that many transport vesicles from the endoplasmic reticulum are able to move towards the Golgi apparatus, where one of the major things that it does is modify some of its contents. So it is the center of manufacturing, warehousing, sorting, and shipping. It is especially extensive in cells, the Golgi apparatus in particular, which are specialized for secretion. Why this is important is because when you're just looking at a cell and you're looking at the biology inside the cell, your vision can get a little narrow-minded. What you need to understand that a cell is not always functioning in solidarity. It is part of a tissue. It is part of many different types of cells which are functioning um, in concordance with each other for that organ to do its job. So there's a lot that goes into a cell functioning the way it does. And it is especially said to be extensive when we say that it's extensive in cells that are specialized for secretion. If you take any tissue, for example, it has some cells which are, which are just secretory in nature, some which are just glandular, some which are performing some sort of muscular function. So there are different cells which are specialized for different functions within the same tissue. So when it is secretory in nature, those cells have a higher or more abundant Golgi or more extensive Golgi apparatus within those cells. So that's what we see here. Then the Golgi also has kind of a similar architecture, not the same, but a similar architecture where it has, like you can see here, it looks like a stacked pita bread or stack of naan, where it has multiple flattened stacks which are right one next to the other. And the membrane of each cisterna, it separates its internal space from the cytosol. So the membrane is the boundary between the internal environment of the Golgi from the external cytosolic environment. So one side of the Golgi, the cis side, so we have cis medial trans. When you look at the Golgi, if you think about a stack of parathas, um, okay. So imagine that you have a stack of like parathas on your plate. So it has an orientation as well, right? There is one side of the stack that is facing the plate, the other side is facing, the rest of the environment. Similar to that, the side of the Golgi or the side of the entire Golgi apparatus which faces the nucleus is said to be the cis side. The middle part, somewhere in the middle of the stack, is the medial, and the one which is facing the membrane is the trans. So cis, medial, and trans Golgi is forming the entire Golgi network that we have been talking about extensively from before. So the cis side, obviously the side which faces the nucleus is the one which receives material by fusing with vesicle. The other side is the trans side which butts off vesicle from where it transfers or from there the vesicles can travel to other sites inside the cell. Then I think there was a GM130 staining of the Golgi. So this was a staining of the Golgi that was done. A very beautiful video was there to show how it exactly occurs. And here you can see a more sort of 3D arrangement of the same thing. You have the cisternae from which you have the cis or the receiving side of the Golgi apparatus. You have the medial and the finally the trans side, which is the shipping side of the Golgi apparatus. And you can actually see an electron microscopy image of the same as well. So during their transit from the cis to the trans side, products from the ER get modified to reach their final state. So what you got from the ER undergoes modification during this entire transport process. What this includes are modifications of the oligosaccharide portion of the glycoproteins. So glycoproteins, while they have a sugar entity attached to them, those sugar entities or that sugar entity also undergoes several modifications based on what is its job or where it has to go when it reaches the Golgi. This is another separate area of study, not getting into details of it, but each of these compartments are doing their own jobs, and any error in any of them can lead to huge, very drastic consequences for the cell and for the individual at large. So the Golgi can also manufacture its own macromolecules, which includes pectin, and it also includes other non-cellulose polysaccharides. During processing, material moves from cisterna to cisterna, each with its own set of enzymes. So the cis, medial, and trans Golgi are just not segregated based on um, how they're arranged or whether they're facing the nucleus, facing the plasma membrane. No, not in that sense only, but also because of the type of job that they carry out. Each has its own each has a different composition. Each has its own job, and therefore each of them have their own set of enzymes. Finally, what the Golgi does is that it tags the protein appropriately. It sorts it where it needs, based on where it needs to go, and then it packages those materials into the transport vessel. So that is the entire composite job of the Golgi apparatus. Here we had a little bit of insight into the fact that the Golgi is also quite intricately associated with the cytoskeletal elements within the cell. So we have microtubules, which are also some microtubules are actually said to be Golgi anchored or they are Golgi associated. So what you see here, it's a sort of drawn schematic. So what you see here is that there is the ER yellow in yellow color. I don't my cursor will be visible here, but yes, this is yellow in color. That's the ER. Following which we have the green colored Golgi. We have the red actin filaments at the leading edge of the cell, which is because the cell looks like it's moving probably in one direction. And we have microtubules, which are sort of purplish in color, which are derived from the Golgi complex. So these are Golgi-derived microtubules, versus there are some blue in color, which are derived from the centrioles, which are present somewhere here. If you can see, from there they have originated. And you have secretory vesicles, which move along the microtubules of the Golgi complex. So those are specifically moving. Some vesicles, they are not just getting pinched in and out of they're not just getting pinched in and out of the Golgi network, but instead there are in some cases, they're actually associated to a skeletal structure in order to direct their proper movement, particularly for some types of cells. If you take an example, the best example would be neurons, which have huge distances to cover. And therefore, you need to have some sort of setup in place which would carry those cargo to the correct destination. So secretory vesicles in some cases are moving along microtubules of the Golgi complex, and in the ones that you see in red, they are moving along the centrosomal microtubules based on what is the cargo, where it needs to go, and who is mediating its transport. The cytoskeletal element or the type of cytoskeletal element that is associated with it would also or could also vary. So that's what we see in this figure. And here there was an example of the fact that you are seeing a cell that is being labeled with some sort of a surface marker. Any cell surface marker can show you that when a cell is detached. Under normal circumstances, there are some sort of cells, because I work with cancer cells, I'll give you those examples. So most of them are adherent in nature. So when they are adherent, they take up a distinct morphology, like some sort of polygonal shape. Each type of cell or different types of cell lines can take up different types of orientation as well, or different types of morphology as well. But when you trypsinize them, which is that you detach them from their attached state, what happens is that they immediately take up a spherical conformation. And that is what this also looks like. It looks when it is detached, it takes up this sort of circular, or not circular, in 3 dimensions, a spherical sort of structure, and then it can re-adhere to the surface. Then it will take up that distinct morphology again. Now, in each of these states, what happens to the Golgi? You are seeing that the Golgi also, it is taking up different sort of arrangements in each case. So the trans Golgi is actually breaking and then gets distributed inside the cell. You see that dotted boundary, and within that, you're seeing that there is a rearrangement of the Golgi that has occurred. When the cells again adheres, then you are seeing that the trans Golgi is reorganizing, and the green line shows that it is getting attached to a matrix protein. So upon attachment, the reorganization is occurring. So inside the cell, the trans Golgi also undergoes resuspension and rearrangement based on which state it is in. Now, however, we see that the loss of adhesion does not affect actin and microtubules. That is, the cytoskeletal elements by themselves do not get affected. So these are the different conditions in which first when the cell, so like I was saying that, cells take up a distinct morphology under normal circumstances when they're adhered. So when they're stably adhered, you are seeing that these are the different types of arrangements you are seeing for actin and for microtubules, versus when they are suspended at 0th minute, suspended after 90 minutes, you are seeing different types of arrangements that are occurring. Here we see the 2 types of microtubules that we just talked about before as well, some of which are centrosome nucleated. They function, their job mainly is in cell migration, and they do so by regulatory effects on centrosome positioning. Versus when we talk about the Golgi nucleated microtubules, then those might function in cell migration, but not through the effects on centrosome positioning. And this we see is the direction of migration, and this would be the leading edge in those cases. So here is the Golgi apparatus, the 2 models of trafficking that we see here. The first is the cisternal maturation model. In this case, as a new cis cisterna is formed, so here is your cis state, the nucleus would be on this side, considered to be on this side, and these are the cis medial and trans Golgi that we talked about. So when cis cisterna is formed, it traverses the entire Golgi stack and changes as it matures by accumulating medial and then trans enzymes through the vesicles that move from later to earlier cisternae. There is a retrograde direction of movement that is believed to be occurring. So this is the cisternal maturation model, that as it moves through the cisterna, it acquires the different enzymes that are components of each of these compartments, and it moves through the entire entire space. Versus if you talk about the other maturation model, the vesicular transport model, both of these are being explored. The one I talked about was the vesicular transport model, that vesicles are moving from cis to medial to trans. They're getting pitched off from one, they're fusing to the next, they're getting pitched off from the next, and they're fusing to the third in that manner. And from there, they are getting pitched off and going to the plasma membrane or to wherever the destined site is. So here, each cisterna remains in one place with no change in enzymes, and the proteins are moving forward through the stack via vesicles that move from earlier to later cisterna. So they believe in anterograde means forward movement and retrograde means backward movement. So these are the two types of models. Again, we do not have one comprehensive, conclusive answer for this. There are studies that have been taken up in both directions. So, when you start off, you take multiple hypotheses, or you at least one group starts off with one hypothesis and they try to sort of provide experimental evidence that the model they are proposing is correct. And then the other groups which are proposing a different hypothesis will try to provide evidence in support or um, debating the same. So it depends upon what eventually gets proven sometime in the future. So those were the two models that we talked about. And here you can see the type of modifications that we're talking about, the glycosylation that is occurring within the ER and the Golgi. So from the ER, we are seeing that there is a UDP and there is an attachment of the mannose glucanac molecules. So not getting into details, but basically these are all sugar moieties. You're seeing mannose, galactose, and glucanac or N-acetyl glucosamine that gets attached in the ER lumen. And from there, as it moves towards the cis medial and trans Golgi, there is some snipping which is done. Some specific residues are removed, and after all of these modifications are made, it is directed to the correct site. So a lot of things actually take place. This is a very extensive study, very extensive chapter, you can think of it, that needs to be studied. But anyway, from here, you get the basic idea of what is happening. So we saw that the proteins can get synthesized in the ER or in the cytosol. From there, they move through the cis medial trans Golgi. From there, some of those become secretory in nature and then pooped out of the cell. Some of them enter the endosomes, and the endosome and lysosome are said to flip, or they have some sort of exchange or equilibrium between these two structures, and they can otherwise get directed to wherever the correct destined site might be. And this was an example of it is the molecule. It's a space filling model at the end of the day, but it is the molecular anatomy of what an organelle looks like. It's not just a beautiful sphere that is there. That would be quite pointless and quite risky for the cell to have. So it has a large number of proteins on the surface, and that is what you see here. You see so many molecules. Like there are SNAP29, there's synaptophysin, there are ATPases, synaptobrevins, syntaxin, glutamate receptors, trimeric GTPases, transporters, etc. And they all are extremely crucial for them to do something as simple as what we are just uttering as, pinching off infusion. How is that happening? That needs some specific proteins that are carrying out those jobs. There are some proteins which bring these two membranes close together and then they allow that fusion to take place. There are some which are sort of if you've ever had like a small patch of a balloon and you sort of try to inflate that and then you twist it around. So sort of that twisting, or not necessarily twisting, but analogous to that, if you have to twist and snip and remove that part of the vesicle, or if you just think about, just dough, when somebody is making, say, rotis, for example, and you have an entire dough, from there you pinch off that vesicle. How is that pinching occurring? There are some proteins which are doing that. Some of those proteins are here. So there's a coordination between a large number of proteins which carry that out. So these vesicles, they emerge with some of those proteins, some of them remain behind from the organelle that they have emerged from. So these are extremely detailed, but if you just see the cross section, it would give you an idea that how intricately controlled this entire process is. And in the third figure, we are just looking at synaptobrevin to show that how abundant it is on the cell surface membrane, and how densely it is populating the entire membrane surface. So that gives you the idea. And I think there was one more video about protein packing that showed how this entire process of trafficking was carried out and what are the different types of proteins that participate in it and how it is regulated, etc., etc. So after that, we come to the lysosome and the vacuoles basically, and that would cover up the topics that were discussed for that week. So just give me a minute and we'll get right to it. Okay. Um, yeah, so we were talking about the lysosome. So yeah, we talked about the other compartments, the ER and Golgi mainly before this. Now coming to the remaining endomembranous compartments. First is the lysosome, and of course it's a membrane-bound sac of hydrolytic enzymes that digests the macromolecules. So what we see here as the basic idea of a lysosome is that it is the suicidal bag of the cell, or that it is the digestive bag of the cell, that whatever goes inside, it's just it's killed by the cell. So that is where all of the digestion occurs. I think we have discussed this previously as well. So it's that membrane bound sac which is very, very rich in hydrolytic enzymes, and it has an acidic pH, as you can see here as well, that it has a pH of approximately 5.0. And the type of enzymes that are present within the lysozyme include acid hydrolase, of course, because of which the pH is quite critical. And apart from that, we have nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, and phospholipases as well. And you can see that the dimension of the lysosome is somewhere around 0.2, 0.5 microns, and it is quite enriched in a proton pump. The reason being that the pH that I talked about, which is so crucial for the lysozyme to function, or for the lysosome not lysozyme, the lysosome to function, because it needs to maintain that pH. And the basic idea of pH, as we know, it is kind of dependent upon the proton or the concentration of H plus ion, so the proton concentration inside the compartment that we're talking about. So if it is H plus, then there has to be an active pump. Um, Active in the sense that if necessary, it would require an energy investment, and that in this case that would be ATP, that would actively push protons that are present inside the cytosol right back into the cell. So for that, we need to have such a pump in place. So that is what we see here. And you can see all of these little black colored compartments, which are the lysozymes that are present, lysosomes that are present inside the white blood cell. So it is the lysosomal membrane that we talked about. It is protected from the acid hydrolases by the lysosome specific expression of some membrane proteins like LAMP1 and LAMP2, which are very, very heavily glycosylated, and therefore they are capable of resisting digestion. So the glycosylation that we were talking about is the job of the ER and the Golgi to correctly glycosylate proteins when it is required. It also has a major role to play because in the upcoming movement that is occurring, it is capable of resisting digestion quite well. So the acid hydrolases are hydrolytic enzymes that are active under acidic condition. Therefore, the lysosomal lumen is maintained at an acidic pH by a proton ATPase in the membrane that pumps H plus into the lysosome. So that's what we see there. And they are capable of hydrolyzing or breaking down proteins, fats, polysaccharides, and of course, nucleic acids. These enzymes, they are capable of working best at a pH of approximately 5. And therefore, the proteins in the lysosomal membrane are capable of pumping hydrogen ions from the cytosol into the lysosomal lumen. While rupturing one or a few lysosomes has very little impact on a cell, massive leakage from lysosomes can destroy a cell by autodigestion. So if all of the lysosomes within a cell underwent rupturing, all of these enzymes would we would be released into the cellular environment. And while the pH is not very conducive for it, you have to understand that the H plus ion concentration, while it is diluted, these enzymes can still perform, while maybe not at the best of their abilities, but they can still perform and they can still carry out autodigestion of important components of the cell, important proteins of the cell, important lipids of the cell. And they can rupture the cellular membrane and they can release all of the cellular contents into the outside and can completely destroy the cell by and large. So the lysosomes basically allow the creation of a restricted space within which the cell is ensuring that whatever degradation or whatever digestion needs to be carried out is carried out only within that compartment so that the cell is not adversely affected under normal circumstances. So it is maintaining that boundary between the lysosomal environment and the rest of the cell. So that is what we see here, that the lysosomal enzymes and membrane are synthesized by the rough endoplasmic reticulum, and then they are transferred to the Golgi. And at least some lysosomes, they bud from the trans phase of the Golgi. So this is the flow that we were already looking at, that from the rough ER transport vesicles goes to the Golgi. From the Golgi, we are seeing that two types of vesicles are emerging. One type is said to go into the lysosomal compartment, and that can either go for digestion or it can go to function as a food vacuole. Then that food vacuole is going to the external of the cell, and whatever food is supposed to be released, that is getting released into the external environment. On the other hand, there is another process known as autophagy, where basically degradation is occurring. So there is a fusion between a phagosome and the vesicle that is coming, and then digestion is occurring. So whatever compartments of the cell are supposed to be digested are getting digested under those circumstances to produce energy. So we'll come to autophagy here. I think, yeah, you can go through Albert's. It's a very detailed process. But basically what is happening here is that when there is engulfment of a bacterial cell or any pathogen, in this case, it is a bacterium, it is getting phagocytosed or localized into the cell. It forms a phagosome. Then that phagosome can fuse with the lysosome and form an phagolysosome, which can then undergo digestion. And what it eventually will do is it will kill the cell. When this is happening to the cell's own compartments, that say mitochondria or some sort of proteins, which are components of the cell that the cell under normal circumstances would protect at any cost, it starts digesting these compartments just so that the cell can produce energy. Then that is called as autophagy, which means self-digestion or self-killing or self-destruction. That self-destruction arises because the cell needs to survive, and it needs to survive at the cost of some of its own components. Not anything that will not allow the cell to survive, but anything that can be dispensed of by the cell is dispensed of by the cell just to survive in the long run. So that angle is a little bit of looking at the greater good, that you are digesting XYZ proteins so that the cell can survive X number of days more. So it's a very fine-tuned process, and autophagy has both some positive roles in context of cancer. I mean, it has some positive roles, some negative roles. You can read up on it. It's a slightly complicated concept, but very beautiful and very um, easy to confuse with apoptosis, but they are not the same. They are very different processes, very differently regulated, and operational under very different circumstances. So you can read up on it, and you can get back to us if you want any of these processes explained in more detail. Uh, Right. So the lysosomes can fuse with food vacuoles, which are formed when a food item is brought into the cell by phagocytosis. And as the polymers are digested, their monomers will pass out to the cytosol to provide nutrition to the cell. On the other hand, lysosomes can also fuse with some cellular compartments, some organelle or some part of the cytosol. And this recycling of the cell's own compartments, which are undergoing digestion, is called as autophagy. What it effectively does is that it renews the cell and provides the cell some sort of um, energy source for it to persevere through whatever difficult times it is going through. So this you see is the peroxisomal fragment and the mitochondrial fragment, which is present within the lysosome. That is sort of a snapshot of how autophagy is occurring. So autophagy basically is the process of cell digestion. So in that case, the autophagosome can fuse or might fuse with an endosome, which is the product of endocytosis. And this is a form of heterophagy, when the cell is internalizing and degrading material that is originating outside the cell. On the other hand, autophagy is cell digestion. So the cell is consuming some part of it, it's some part of its own self, and it is producing energy for its own self to carry out. The concept of autophagy received its the reason why I'm telling you it's such a beautiful and complicated concept is that it was an immense recognition, and it did receive the honor it deserves as the Nobel Prize in Physiology and Medicine in 2016. Also, if anybody is preparing for any competitive exams, keep in mind the scientists and the work that they have done that has been awarded Nobel Prize this year. And of course, the last few years as well, in particular for this year, because it was so relevant to the pandemic that we have just all encountered and what we have gotten through. So the prize that was awarded to Kathleen Fresco, and the concept of mRNA vaccines, why it was so revolutionary, how did a work that was so recent receive such immense recognition so fast? Because usually concepts or research ideas or discoveries that receive Nobel Prizes, they are given that timeline to be defined as a remarkable contribution to mankind, and only then they are awarded. But considering the immense impact that the idea of mRNA vaccines had in that short period of time, in those 2 years, 2020, 2021, majorly, the quick action of all of these scientists, the application of it, and the huge impact that it had on the population by and large, it brought forth the subtle use of concepts that can bring immense change to mankind. So that impact gets recognized, and that is why it got the recognition that it did, and it got the Nobel that it did in such a short duration. So these are all remarkable discoveries in the field of science. So you can keep these in mind. You can keep in mind the concept of mRNA vaccines, how they're applicable, which companies actually brought up these concepts as the mRNA vaccines were released. Because not all did. If you remember, there were different types of vaccines. There were COVID, there was Covishield, there was Covaxin, and they all had different biological concepts that operated in those cases. So keep in mind these ideas, because these are recent events, and these are recent events that have occurred in academia and research as well. So they would be relevant to for your own knowledge and for any exams that you might be preparing for in your master's or for your PhD in the future if you're interested. So keep in mind these examples for sure. And Dr. Yoshinori Oshumi, he was awarded the Nobel Prize in 2016 for the concept of autophagy that was studied by him and his group. So yeah, we have talked about autophagy, and we've seen that how one part of it, which is what I have majorly talked about till now, is the starvation-induced autophagy, which is non-selective, which is where the cell does not have any energy, and it needs to survive no matter what's the cost, and therefore what it chooses is to degrade anything and everything that it will get, and it will degrade cytoplasmic components, it will degrade organelles, it will form sort of a isolation membrane, form an autophagosome, will degrade it, and will produce energy. On the other hand, when we talk about selective autophagy, only certain components inside the cell will undergo this entire process or will undergo digestion under such circumstances, and therefore the cell cherry picks what it wants to degrade or what it can afford to degrade under those circumstances and will produce energy from those. So that is the concept of autophagy that we have talked about. And the term itself, it comes from the Greek, its etymology lies with the Greek origin, which is meaning of self-eating. So autophagy is an evolutionarily conserved process by which cellular cargo or cytoplasmic cargo is sequestered inside double membranous vesicles that are delivered to the lysosome for degradation. And autophagy substrates, they include organelles like mitochondria, aggregate-prone proteins that cause neurodegeneration, and various other proteins as well. Uh, in the early, so we are going back to the idea that was there. So in the 1950s, Christian de Duve, using cell-fractionation methods developed by Albert Claude, found that proteolytic enzymes were sequestered within a previously unknown membrane structure that they named, or that was coined as the lysosome by them because it was lysing the components inside the cell. And Christian de Duve, Albert Claude, and George Ballard, you can actually read. There are a lot of discoveries by Ballard that you can actually read up on, and they're fantastic. I mean, of course, all of these scientists have some fantastic contributions, but particularly for autophagy, you can look at Ballard's groups, the work of George Ballard. So they were awarded the Nobel Prize in Physiology and Medicine in 1974 for their discoveries concerning the structural and functional organization of the cell. So everything that you and me do today and study today with so much ease and so much obviousness to the understanding that we have today has been the landmark contribution of all of these immensely talented and intellectually supreme scientists that have seen something, observed something, and have drawn some beautiful conclusions from it, and that have given us the concepts that we understand today and that we take for granted today. So, somebody might have just looked at it as some globule, as some structure, and would have ignored it, but they didn't. They looked into it and they saw that this might be something different. It might have some specific function within the cell, and look at the kind of functions that are being discovered today, and so much is still not known. So every day in science is a new day to discover something new. So just trying to drop some inspiration because research, while exhausting, can be life altering, not just for an individual, but for mankind at large. So coming back to the content that we see here, Christian de Duve, he also found portions of the cytoplasm to be sequestered into the membranous structures during normal kidney development. And he recognized that the structures had the capacity to digest part of the intracellular content. And he termed the concept of autophagy in 1963, that is self-digestion for long-term renewal. Further, in the early 1990s, Yoshinori Oshimi, he decided to study autophagy using the budding yeast Saccharomyces cerevisiae as the model system. And the yeast vacuole he found is the functional equivalent of the mammalian lysosome. So he used a different organism, but the concept was elucidated by him. He found that autophagic bodies, that is the autophagosome and other similar membranous structures, which are actually carrying out the digestion that we are talking about today, they accumulate in the vacuole when the engineered yeasts were grown in a nutrient-deprived medium. That is when you're subjecting them to starvation pressure. Then these organisms are overcoming that stress or that pressure by carrying out these sort of processes. So that is what we see there. And this has a detailed understanding of how the process occurs. I'm not getting into it. You can read it for your own understanding. But the basic steps are that first there is induction of autophagy because of some stimuli. Either there's nutrient scarcity, low oxygen, growth factors absent, so on and so forth. Then there is nucleation where the phagophore it forms, and then there is expansion. The cargo is recognized, which needs to be sequestered. Then comes the process of expansion and cargo recognition, continuing further on. And we see that there is protein recycling and fusion, which is occurring. Then there is the digestion, fusion between the autophagosome with the lysosome. And then the digestion process has to occur, where within the autolysosome, enzymes would gain access to the cargo, would break it down. And finally, the cargo that has been produced would be recycled within the cell. When we say cargo, we mean the basic macromolecules or the basic monomers that you need, not macromolecules, monomers that you need for cell survival, which majorly includes amino acids for new proteins to be synthesized and for the cell to function. So that cargo would get recycled within the cell and would be released into the cytoplasm for cell to do whatever jobs it needs to do to survive. So that is the by and large idea of what happens during autophagy, what happens and why does it happen? Why is it necessary? Why does the cell at all choose to digest itself? These are all questions that have been understood to some extent. But yeah, you can look it up at any time if you want more detailed insight into it. Uh, Next, we have the vacuoles. So vesicles and vacuoles, which are larger versions, are membrane-bound sacs with some different types of functions. So we have food vacuoles, which from phagocytosis, they are fusing with the lysosomes. We have contractile vacuoles, which are found in some sort of freshwater protists, which are they are contractile, which means there's some sort of pumping function which is occurring. They pump excess water out of the cell. And then we have central vacuoles, which are found in many mature plant cells as well. So that has a different sort of function. It has it is functioning as the tonoplast for turgidity, a number of other functions. So here is what you see, the contractile vacuole that we talked about. Which is present in several protista. And here we have cilia, the large nucleus, the oral groove, food vacuole, so on and so forth. So the membrane surrounding the central vacuole, which is the tonoplast, is selective in its transport of solutes into the central vacuole. The functions of the central vacuole include stockpiling proteins or inorganic ions, depositing metabolic byproducts, storing pigments, and storing defensive compounds against the herbivores. Also, it increases the surface area to volume ratio for the whole cell, and two different types of plant vacuoles have been identified. The first are protein storage vacuoles of neutral pH, the lytic vacuoles of acidic pH, which are equivalent in function to the lysosomes that we know for mammalian cells. And And then we have the plant vacuole, which can act as a storage organelle for both nutrients and for waste products. The plant vacuole can act as a degradative compartment. It has an economical way of increasing cell size as a controller of turbo pressure, such that the osmotic pressure, it pushes from the cell interior to the outwards and keeps the plant sturdy and intact and does not allow it to unnecessarily wilt. So this is what the central vacuoles sound like, and you can looks like. You can see that in the plant cell, it occupies a major percentage of the cellular area, and that is basically its job. And yeah, that pretty much wraps up all the topics that were part of this week. It might have gone by a little fast, but if there are any doubts, please do go ahead and bring it up. So basically, we looked at all of the types of endomembranous structures that are present within the cell, with major focus on the endoplasmic reticulum, the two types of ER, smooth as well as rough, the individual functions of each of these with respect to protein synthesis, protein function, protein modification, and also the metabolic aspect of it, how it contributes to cellular metabolism. Then we looked at the Golgi, the different models that are present there for the type of movement that occurs through the Golgi, the cisternal maturation model or the vesicular pinching model. Then we looked at the job of the Golgi that it does, the different orientations that it has, and how it heads in the further direction of the protein to its destined compartment. Following this, we also looked at the other two types of endomembranous organelles, mainly the lysosome and its job with respect to autophagy or cell digestion. And certainly we looked at the vacuole as well very briefly. So that majorly brings or compositely shows the key role of the endomembrane system in synthesis and hydrolysis of macromolecules within the cell and how they modify macromolecules for different types of functions. So that sums up our discussion for this week. We will just look at a few sample problems and we'll wrap up the today's session. So for that, just give me a minute's break and we'll be back. So we'll just go with the sample problems for this week's topics. So among those, the first question is that what is the role of Golgi apparatus in cells? First is carbohydrate synthesis, second is lipid metabolism, third is ATP production or processing and sorting of proteins. So like we've just discussed that Golgi is majorly involved in the sorting process after trafficking from the ER. It begins at the ER, but then it is carried on forward by the Golgi. Secondly, we are asked that the Golgi apparatus is a membrane-less structure, is a tubular network, consists of flattened membranous sacs, or is a set of lamin fibers. So those are, if you remember that parking lot model, they are this stack of flattened membranous sacs that are arranged one on top of the other in that helicoid sort of arrangement. So option C is correct. Then in question 3, we're asked that which of the following statements about the Golgi apparatus is not true? The trans phase of the Golgi, it sorts proteins. The microtubules play a major role in organization of the Golgi apparatus. Golgi is a membrane-bound organelle, or it is involved in the regulation of mRNA synthesis. So which of these is not true is the question. So the trans phase sort proteins is it is true. It is yeah, it's I think marked it incorrectly probably. Um yeah, it's incorrectly marked. I'll shift it right now. So Golgi is involved in the regulation of mRNA synthesis. That is certainly not true. It does not play a role in that. It is a membrane-bound organelle, and microtubules do play a major role in the organization of the Golgi apparatus. Uh, then we are asked that in question 4, tunicamycin is an antibiotic that inhibits the synthesis of carbohydrates. Which of the following organellar activity would get affected by tunicamycin? So is it Golgi? Is it mitochondria? Is it an ER or lysosome? So it would be the ER because we have discussed to some extent, we have discussed the glycosylation that occurs. So within the ER, so also within the Golgi, but it initiates in the ER. So we can certainly look at tunicamycin affecting the endoplasmic reticulum. So you can read up on that as well. And also there are some metabolic aspects that we briefly discussed that are associated with the ER. So for your own interest, please go ahead and look at how tunicamycin works and why does it only affect the ER and not the other organelles, or does it have any effect on the other organelles? So that's something you can certainly study for your own better understanding. The fifth question is about how the rough endoplasmic reticulum is abundant in cells that secrete proteins, that synthesize amino acids, that secrete lipids, or that synthesize nucleotides. Among them, the correct answer is that it is abundant in cells that secrete proteins, which is option B. Then in question 6, we are asked that the endoplasmic reticulum, which is associated with ribosomes, is called rough endoplasmic reticulum, smooth endoplasmic reticulum, riboendoplasmic reticulum, or the ER lumen. So it is the RER or the rough endoplasmic reticulum. That is option A. Then lysosomes are called suicide bags of the cell because A, they digest carbohydrates, B, they can carry hydrolytic enzymes which when released into the cytoplasm can kill the cell. They can fuse with endosomes or they can recycle the waste that is generated in the cells. So that is obviously option B is the correct option, that they can carry hydrolytic enzymes that can be released into the cytoplasm. A major component of vacuoles is A, water, B, hydrolytic enzymes, C, acid phosphatases, or D, aldolase. So major component of vacuoles is certainly water. That is option A. Then in question 9, we are asked that which of the following statements about vacuoles is not true. They provide turgidity and rigidity to the plant cells. They help maintain osmotic pressure in the cells. They store toxic metabolic byproducts and thus protect the cells, or they regulate cell division and they help in plant growth. So the option D is certainly not something that vacuoles do. So option D is the correct option, that it does not regulate cell division or help in plant growth. And finally, cell sap that we talk about for plant cells is often the cytoplasm of plant cells. It is the fluid in plant vacuoles, the fluid in nucleus of plant cells, or the lumen of plant endoplasmic reticulum. So the correct option for this case is that it is indeed the fluid that is present in plant vacuoles. So these were just included. Yeah, these are week 7 sample problems. That it was some confusion from my end. I'll just edit that. So I think with this, we are basically summing up our discussion for today. Let me just a little bit. Yeah. So basically today we looked at the topics that are covered for this week, for week 7. And we looked at some sample problems and we looked at some of the questions that were present or the assignment questions that were part of the previous week. And apart from that, we have discussed the types of endomembranous systems, each of the different organelles, their function, and how their function can differ based on what is the condition of the cell, like the lysosome, which normally performs digestion of components that are supposed to undergo selective digestion. However, under dire circumstances, it can digest the entire cell just to produce energy in order for the cell to survive properly. So those were all of the different topics that were covered. Finally, we have ended with some sample problems, and we will look at more questions for the upcoming week. And the next session will be on Sunday. So if you have any doubts, please bring them in. That will be the last session that I will be conducting from my end for this course. And you can bring in your doubts if you have any. You can bring in any questions. If you want something that is to be explained, I'll try to sort that out as well in that session. And yeah, I guess we are done for this week. So thank you, and all the best for your exams that lies ahead. So thank you so much, and good night.